Hello everyone, my name is Kashi and welcome to my brand new YouTube channel where I'm going to be making a bunch of videos teaching you guys about all sorts of subjects, biology, chemistry, maybe even some psychology in the future. I love learning and teaching others, so I think that making this channel is going to be a lot of fun. And to start off, I'm going to be making a collection of videos exploring a subject called cell signaling. I just finished taking a course all about cell signaling in my first year of uni, and I thought it was really interesting. Also kind of complicated though, but I'm hoping that these videos would be a great way for me to share my knowledge in the subject. So let's get started. Alright, so first let's just review what we're going to talk about in this video. So first we're going to look at what cell signaling actually means and what even is it. Then we're going to talk about some of the different common types of cell signaling. And then, this is the important part, we're going to look at a general framework that my professors taught me and I found really useful. And what's cool is that this framework for cell signaling can later be applied to a bunch of different molecules that we can explore in future videos, such as histamine, norepinephrine, acetylcholine, maybe different types of steroids or prostaglandins, and many more. And that's going to help us in the future. Okay, so what even is cell signaling? Cell signaling, it's actually pretty simple. It's just how cells talk to each other in order to produce a response. That's all. So a common way that it can happen is this. So you have cell A, which is like your parent cell, and then you have cell B, which is like your target cell. So the parent cell is trying to communicate with the target cell. And one way it can do this is it can produce a messenger molecule. So inside cell A, it makes this messenger molecule that I'm labeling M. And then once this messenger molecule is produced, it can leave cell A and it can make its way over to the target cell. And then in the target cell, it can produce a response. That's about it. Now, this is usually how it usually happens, but not always. The annoying thing about cell signaling is it's hard to set concrete rules because there's always exceptions. So now let's look at some of the common types of ways that cell signaling happens. Alright, so now that we've looked at what cell signaling actually is, let's look at the different types of cell signaling. There's four main ways. So the simplest, is what's called paracrine signaling. And this is what we did in the last slide. It's very simple. You have two cells that are, and this is important. The two cells are very close together. They're extremely close together, like basically right next to each other or like basically in the same area. And you have your first cell, which is going to make a messenger molecule that leaves the cell and it goes outside into the extracellular space, which is the space outside the cells. And then it makes its way over to the other cell and it'll bind to a receptor or something and it will produce a response in the neighboring cell. So very simple, one cell makes a messenger, goes and does a response in the second cell, and the two cells are close by. Now, autocrine signaling is a little bit more weird. This doesn't look like what we saw in the last slide, because autocrine signaling actually only involves one cell instead of two. I know, it's weird, like how are you going to signal with one cell? Well, it's actually just because that one cell talks to itself. So that one cell is going to make a messenger molecule, just like in the other one. And that messenger molecule is going to leave the cell, and then it's just going to bind onto a receptor of that OG same cell, and it's just going to make a response in that same cell. So the cell is literally just talking to itself. It just makes a messenger that comes and binds to the same cell and produces a response. So then we have synaptic. Oh, something happened to my drawing. Alright, so then we have synaptic signaling. Now this one is a lot more complicated and I'm going to do like a much more deep dive into synaptic signaling in a future video, so don't worry about that one for now. But the general premise is it's similar to paracrine except it's between two different neurons, or like a neuron and a muscle, and it's over this tiny gap which is called a synapse, hence the name synaptic signaling. And like a messenger will go from one neuron to the other, but in reality it's a lot more complicated, so don't worry about it for now. But if you notice, there's a common theme with autocrine, paracrine, and synaptic signaling. All three of these signaling types, the messenger molecule just has to travel over a very short distance. Like in autocrine, it just goes outside, then it comes right back in. Paracrine, it just goes outside, then it goes and talks to a neighboring cell, and then synaptic signaling, it just has to travel across a really tiny synapse. But endocrine signaling, on the other hand, it's similar to paracrine. It's one cell talking to another, but the difference is these cells are very far apart. So far apart that the messenger molecule that's produced by the first cell, it goes outside and it actually has to travel through the blood to get to the next cell and produce a response. And there's a special name for these messenger molecules. They are known as hormones. You probably heard of them before. Hormones they're just messenger molecules that go from one cell to another, but they're specific to the endocrine system. They have to travel through the blood, and they have to go for much further distances. And an important quality of hormones is that they are stable, because with autocrine signaling, paracrine signaling, synaptic signaling, because it's very short distances, these messenger molecules are pretty unstable in the sense that they get broken down by the body pretty fast. And that's okay, because it's just going over to a neighboring cell. Like, who cares if it gets broken down fast? Like, it's going to make it to the neighboring cell and produce response quick enough before it gets degraded. But with endocrine signaling, hormones need to be a lot more stable because they're traveling for much longer distances. They have to be able to travel much longer and much further without getting broken down. So hormones are a lot more stable and strong compared to our other messenger molecules. And yeah, that's about it. Those are our 4 main types of cell signaling. So now let's get into our framework. Okay, so for the cell signaling framework, my professors taught me a really useful set of letters to remember it. And this is PPRRT. So the first P stands for production. You got to produce the messenger molecule. The next one is packaging. Sometimes the messenger molecule will have to be packaged and stored, whereas other times it's not stored. And then there is release. So the messenger molecule must be released from the first cell and make its way over to the second cell, where it will produce a response. And then comes termination. We need to terminate the signal to prevent it from going on indefinitely. So now let's explore all of these steps in depth one by one, starting with production. This is probably the easiest one. Alright, so the first step, pretty simple, pretty self-explanatory. It's production. So the messenger molecule needs to be produced. So if you have your parent cell and then you have your target cell, the messenger molecule needs to be produced or synthesized in your parent cell before it can leave and go do its stuff in the target cell. It needs to be made. That's about it. And I mean, you can remember it as parent cell produces, PP, facts, or you can remember parents produce children. So parent cell produces the messenger molecule. This one's pretty self-explanatory though. The next step is packaging. This one's a little bit more funky. Alright, so for the next step, packaging, you can see I have this little question mark here, right? And that is basically because packaging sometimes happens and it sometimes doesn't. It depends on the messenger molecule. So sometimes it's packaged, and let's look at what happens. So you have your parent you have your parent cell, right? And that's going to produce your messenger molecule. But that messenger molecule, it's not just going to immediately be released. It's going to be kept inside this cell. It's going to be stored, saved for when it's really, really needed. So that and then when an event happens, right? This event basically triggers the release of this messenger molecule. And this event could be a bunch of different things. It could be an allergic reaction. It could be a fight or flight response. It could be some kind of danger, injury, a disease. It could be a bunch of things. But this event stimulates the release. Here, let me write that down. Event stimulates release of an already stored and packaged messenger molecule. And then when this messenger molecule is released, it can go to the target cell and it can produce a response as needed. But this does not always happen. Depending on the messenger molecule, sometimes it's not packaged. Sometimes instead what happens is you have your parent cell, right? And then the event happens, right? But in this case, the event does not stimulate the release. The event instead stimulates the event stimulates production. So when this event happens, this event actually causes the parent cell to produce the messenger molecule. So when what's it called? This is called a what's the name for it again? This is called I got it. This is called synthesized on demand. Synthesized on demand. And as the name suggests, when there's a demand, when the event happens, then only the messenger molecule is synthesized. And as a result, it's a lot slower. Because for the first situation, for packaged molecules, the messenger molecule has already been packaged. So when the event happens, then, you have a bunch of it stored up and ready to go. So you can release it all at once and have a good big response. But for molecules that are not packaged, when the event happens, then, I'll do a laser pointer. When you have an event, then that causes this messenger molecule to be produced. So it's slower because you have to wait for this molecule to be produced. And then when it's produced, it's not stored. It's immediately released right after it's produced. And then it can leave the cell and it can go over to your target cell where a response can happen. So you have packaged molecules where it's produced and stored and the event stimulates release. And then you have non-packaged molecules where the event actually stimulates production and it's a lot more slower, and we call it synthesized on demand. So how come some messenger molecules are packaged while others are not packaged? It depends on the properties of the messenger molecule itself. So when comparing packaged versus non-packaged molecules, the biggest characteristic that plays a role is polarity. Polar molecules are lipophobic, meaning they don't like fats, but they're hydrophilic, meaning they do like water. Alright, and then on the other hand, if a molecule is nonpolar, that means it's lipophilic, so it does like fat. It's the opposite. But it's hydrophobic, meaning it does not like water. So because of this, depending on if a molecule is polar or if a messenger molecule is nonpolar, that determines if it's packaged or non-packaged. And here's why. So let's say you have a messenger molecule, right? And that messenger molecule is being stored inside like a cell or a vesicle or a granule, any kind of storage compartment, right? More often than not, this storage compartment will have a membrane outside of it to separate outside from inside. And this membrane is usually made of lipids. Here's the problem though. If there's a lipid membrane, right, that means there's basically a lipid barrier between the inside and the outside. So if this messenger molecule is lipophobic, let's say this messenger molecule is polar, that means it's lipophobic, meaning it doesn't like fat. So then this messenger molecule is not going to be able to escape because it has a big layer of fat to get through. It can't get through this, right? It doesn't like fats. So because of that, polar molecules, because they don't like fats, they are packageable. I don't even know if that's a word, I can't lie. But they can be packaged, and it's because they're able to be contained in this lipid membrane. But what if your molecule is nonpolar? So same situation. I did not mean to do that. Hold on. Alright, so same situation, right? You have your messenger molecule and it's inside some sort of storage compartment with a membrane that's made of lipids, but this time your messenger molecule is nonpolar. This means it's lipophilic, and that means it likes fats. So in this case, what would happen is, if it likes fat, then this lipid layer it's not a barrier, right? It's no challenge because it likes fat. It's able to pass through fats. So then it's able to just slip through no problem. Nothing is stopping it from going through. So as a result, nonpolar molecules cannot be packaged because there's nothing keeping them inside this little vesicle. They'll just slip right through and go outside and they'll leave, right? There's nothing stopping them. So because of that, depending on if a messenger molecule is polar or nonpolar, depends if it can be packaged or not. So let's look at some examples of both. Polar molecules can include, there's acetylcholine, there's norepinephrine, there's histamine. These are polar molecules, and because of that, they are stored. On the other hand, sorry, my handwriting's terrible. On the other hand, you have nonpolar molecules such as steroids or prostaglandins. And because they're nonpolar, they can slip right through a lipid membrane, and so even if you try to store them, you just can't store them. So these are not packaged. And I hope this makes packaged versus non-packaged molecules a little bit more clear. Alright, so the next step, release, is pretty simple. We basically already covered this in the last step of packaging, but the general premise is, so you have your parent molecule, which produces the messenger molecule, and then after it's produced, it needs to get released so that it can go to the target cell and it can do its stuff. And as we said in the last slide, for molecules that are synthesized on demand, then the events will stimulate production, and then once it's produced, it will immediately get released. But for messenger molecules that are stored, then the event will, then what will happen is it'll get produced and it'll be stored, and then the event will stimulate release. But that's about it. Next step would be response. So after your parent molecule, right? After it makes your messenger and it leaves and it goes over to your target cell, it needs to produce the desired response. But how does this happen? Well, let's say this is your target cell, right? This is your target cell, and your messenger is coming in. What usually happens, again, cell signaling is complicated because there's always exceptions, and it's hard to have like a concrete rule. There's lots of different possibilities, right? But basically what usually happens is the messenger will bind onto a receptor. Receptors can be on the cell surface, or receptors can even be inside the cell. And it can bind onto these receptors. It can go and bind onto this one, or it can go inside and bind onto this receptor. And then that produces it undergoes a process, right? And then the outcome of that process is the response, the desired response. Now this process is what is known as transduction. And that is going to be the focus of our next video. Transduction is complicated. For a long time, it was known as the black box. It was basically a mystery. Like, how does a messenger binding onto a receptor produce the response? How can that just happen? Well, they eventually figured it out by the work of some scientists like Sutherland and a few others. And we're going to focus on that in the next episode. But for now, just know that response is when a messenger molecule binds onto a receptor, something happens in this transduction process and it produces a response. Now, we say that there are some receptors that can be located on the outside of the cell or inside of the cell. Usually, receptors that are located on the inside of the cell what will happen is the responses will have something to do with genomics. So it can be, increasing transcription of mRNA or increasing translation to make proteins. Anything that's related to genes, transcription, translation. And as a result, this takes a longer period of time. Whereas usually, receptors that are on the outside cell membrane will do responses that are non-genomic. And these are often a lot faster. Now again, remember, there's always exceptions. It is possible that these membrane outside bound receptors can do genomic responses. It's also possible that these inside receptors can do non-genomic responses. But most of the time, intracellular receptors are linked to genomic responses, and outside membrane-bound receptors are linked to non-genomic responses. Another thing to note, depending on whether the messenger molecule is polar or non-polar, that also affects whether or not the messenger molecule can enter the cell or if it has to bind to the extracellular receptors. If it's polar, then it doesn't like lipids, so it cannot pass this lipid layer. It cannot enter. So if it's a polar messenger molecule, it will most likely bind to extracellular receptors. Whereas if it's a non-polar messenger molecule, then it can pass the lipid bilayer and it can enter the cell and bind onto these intracellular receptors. But if it wants to, it could also bind to extracellular receptors too, as we'll see in future examples. So the last step in the cell signaling pathway is termination. So you have your messenger molecule, which can enter your target cell and it can produce a response. But the thing is, you don't want this response, right? You don't want this response to be like infinite, because that can lead to problems in the body. Your body wants to stay at balance, homeostasis. If there's excess of something, then that's always bad. So you don't want excess response. You need to be able to limit it and control it. You need to be able to terminate the response. And termination can happen in a bunch of different ways. It can inhibit, like let's say this messenger molecules and going and binding onto a receptor. It can block the receptor, it can get rid of the messenger molecule, it can block the transduction pathways inside the cell, or it can just, let's say the response is producing some kind of chemical, it can just degrade that chemical. Termination can happen in a bunch of different ways. And in future videos, as we explore different signaling pathways of different messenger molecules, we will see a bunch of different termination mechanisms. Alright, that just about wraps up the cell signaling framework. But before we go off and end the video, let's recap real quick. So cell signaling is a process where cells talk to each other, and the outcome is they produce a response. And the different types of cell signaling, so you have autocrine signaling, where a cell will produce a messenger molecule that will leave the cell, go outside, and then come back, bind onto the own cell's receptors in order to produce a response in that very same cell. Then you have paracrine signaling, two cells nearby each other that communicate, so one will release a messenger molecule that goes and does a response in the one nearby it. Then you have synaptic, which is very similar to paracrine, except it's across two different neurons, and the messenger molecules travel across a synapse. And then you have endocrine, which uses hormones, which are messenger molecules that are very, very stable because they have to travel over long distances, and it travels through the blood to go from one cell in one part of the body to another cell far away in the other part of the body. Well, it doesn't have to be in two different parts of the body, but you get the idea. It's over a longer distance than the other three. And then finally, you have your cell signaling framework, as we saw. So the messenger molecule must be produced. It is sometimes packaged, so this one is a maybe, depending on the cell, depending on polarity, the messenger molecule. It may be packaged, it may be not, but after it's produced, or after the event comes and stimulates release, the messenger molecule is released, goes to the target cell, and it does the response. But we don't want excess response, as this can be dangerous, so eventually the response must be terminated in some way or another. And that just about wraps up this first video. I hope this video was able to help you guys understand cell signaling a little bit more, and I'm hoping that this can be a foundation for future videos where we can apply this cell signaling framework to all sorts of molecules like histamines, prostaglandins, acetylcholine, and many more. I'm excited for this channel, and if you guys enjoyed this first video, please consider dropping a like or subscribing. If there's anything you think I need to work on or improve, I'm sure there's lots. This is just my first video. I'm sure that there's lots of places where I messed up, but please drop any feedback, suggestions down in the comment section. I'd really, really appreciate it. But thank you guys so much for watching, and I'll see y'all next time.